Jonathan Becker, 7/23/2025. Today I'm talking about prokaryotic and eukaryotic interactions in the body. So one of the most essential and well-researched examples of prokaryotic and eukaryotic interactions occurs between gut microbiota and human intestinal epithelial cells. The human gastrointestinal tract harbors an estimated 100 million microbial cells, which often outnumber human cells, and form a highly diverse microbial community. This symbiotic relationship is mutualistic, meaning both organisms benefit. The bacteria gain a stable environment and nutrients, while the host gains metabolic support, immune regulation, and protect against harmful microbes. So how do they interact? There's three parts that I talked about. The first part is attachment and molecular signaling. Bacteroides fragilis, a gram-negative anaerobe that resides in the mucus layer of the large intestine, produces polysaccharide A, which binds to TLR2 receptors in dendritic cells in the gut lining. This interaction signals the immune system to produce regulatory T cells and release anti-inflammatory cytokines like IL-10, helping to reduce gut inflammation and prevent diseases like colitis. Second one is immune modulation. Polysaccharide A helps keep the immune system in balance by increasing Treg cells and suppressing Th17 cells, which are linked to inflammation. This prevents overreactions to harmless microbes or food particles and supports immune tolerance in the gut. Additionally, bacteria release short-chain fatty acids, SCFAs, like butyrate, which help maintain intestinal barrier integrity, reduce inflammation, and fuel epithelial cells, especially colonocytes. SCFAs and support. Other gut bacteria ferment fiber into short-chain fatty acids like butyrate, which provide energy to intestinal cells, and they strengthen the gut barrier and support immune balance by encouraging more Treg production through receptors like GPR43. And then this is a diagram. So right here we have the Bacteroides fragilis, and then right here is the eukaryotic intestinal epithelial cell. And so here I've labeled all the parts. This right here is the DNA. This is the inner membrane, the outer membrane, the pili, and the flagellum. So clearly it's prokaryotic because it doesn't have a nucleus. And so what it does is the LPS, which is the lipopolysaccharides, are recognized by the TLR4, the toll-like receptor, on the surface of the eukaryotic cell. This is a key immune detection event. Then the TLR4 activation triggers an immune response in the eukaryotic cell. Signals are sent inside the cell into the cytoplasm and potentially the nucleus. And this may lead to the production of inflammatory molecules or antimicrobial peptides to fight infections. So like I was saying, LPS is a molecule found on the outer membrane of many gram-negative bacteria. It acts as a danger signal to your body, kind of like a red flag. The eukaryotic cell detects the lipopolysaccharide through the TLR4, and the TLR4 is like a sensor or security alarm built into the cell membrane. When LPs bind to TLR4s, it activates the receptor. So then the TLR4 sends a signal inside the cell when it binds, and it's like flipping a switch that turns on a bunch of internal machinery. The cell responds by activating genes, so the signal travels to the nucleus of the eukaryotic cell, and there it activates genes that produce cytokines, defensins, and other inflammatory molecules, and then the immune system joins the fight. So nearby immune cells may be called in to swallow and destroy the bacteria or ramp up inflammation. And then lastly, here are my sources that I use. And that's it. Thank you guys for listening.